with the conference, so if people could take their seats, please. So you're all very welcome back, full of coffee and ready to go, I hope. During this section, we'll be hearing about updates in science and technology. And again, to remind you that Slido is there, so if during any of the presentations questions springs to mind, put it into Slido and we will do our best to get through as many questions as possible. So first up, I would like to invite Minister of State with special responsibility for research, Noel Grealish, to say a few words.
Thank you. Ladies and gentlemen, good morning. It's a pleasure to welcome you all here this morning to the Agricultural and Climate Change Science into Action Conference here in this beautiful area of Dublin Castle. I'm heartened by the strong turnout today, which reflects the commitment of stakeholders across the agri-food sector. Your presence underscores a shared determination to address the environmental and climate challenges facing our sector and to deliver tangible, positive outcomes. Today marks an important step forward as we move from discussion to action in our collective response to climate change. The upcoming sessions represent the very heart of this conference, applying science to the real-world challenges. We will hear from leading Irish and international researchers working on emissions reduction, soil and nutrient management, livestock genetics, food technologies, land use and knowledge transfer. These sessions provide us with a unique insight into current research, tackling emissions of carbon dioxide, methane and nitrous oxide arising from agricultural practice and land use. Let me be clear, this is not research for researchers' sake. This is practical, applied, farmer-focused science. It also recognises fundamental research without which the foundations for applied solutions would not be present. It supports our climate action plan, informs our CAP strategic plan and enables a just transition, one that delivers environmental sustainability while protecting the livelihoods of farm families. The first session today will focus on nitrous oxide, a greenhouse gas that accounts for 22% of Ireland's agricultural emissions. We will explore advances in precision nitrate management, the use of environmental inhibitors and the role of Sward Diversity in mitigating these greenhouse gases. The BioS4 grass project video and the presenters will share important findings on how soils, fertilisers and plant interact, offering tools and strategies to reduce emissions while maintaining or even improving productivity. The next session delegates after that will shift our focus to methane, which accounts for 75% of agricultural emissions in Ireland. This session showcases the depth of our research effort from cutting-edge work on ruminant gut function breeding for low-emission livestock to feed additives and optimising animal performance. Here, international collaboration is at its finest, with contributions from researchers in Ireland, Northern Ireland, New Zealand and Spain. Finally, Session 6 will examine land sinks and sources of carbon and increasingly critical area in our climate strategy. We'll look at how both mineral and organic soils, as well as forest, can sequester, store and release carbon, and how, with careful management, carbon storage can contribute to our national climate targets. A common thread runs through all these sessions, science that is applied, collaborative and focused on delivering solutions. To close this segment of the conference, we'll hear about the real-world application of this research, how it's been translated into practice. This final session will lead into what promises to be a very engaging panel discussion. We'll highlight the systems, people, tools and technologies transforming scientific findings into everyday decision supports on Irish farms. From farmer-led innovation and digital discussion supports to extension services and international knowledge sharing, this session captures the very essence of climate-smart agriculture. In conclusion, delegates, by embracing science, investing in research and innovation and strengthening partnership at home and abroad, we can reshape our agricultural systems to be more climate resilient, economically sustainable and environmentally responsible. My aim is that today's conference will further highlight the groundbreaking research being carried out here in Ireland and with our international partners and the crucial role it plays in driving meaningful change. Finally, I want to acknowledge the dedication of the research community represented here today across our universities, agencies and our international partners. And I want to thank all of you, farmers, advisors, scientists and policy makers, for your commitment to turning knowledge into progress. It is now my great pleasure, delegates, to open the scientific session of the 2025 Agricultural and Climate Change Conference. And this will begin with a short video focused on the research project BioS4 Grass, a department-funded project being led by University College Dublin to look at ways of reducing nitrous oxide emissions using alternative bio-based coatings and fertiliser. And Goramila Mahagiv, and I hope that you enjoyed the rest of the conferences today. Thank you.
A lot of greenhouse gas emissions in the agricultural sector comes from excessive use of nitrogen. Based on estimations, Ireland would need to reduce about 20% the amount of nitrogen being applied on farms. And this is a challenge that inspired this project. So this project is based on the use of biostimulants in grass production. So what we're trying to achieve here is to use biostimulants as a way to reduce the amount of nitrogen fertilizer added onto farms. This project really builds on research that has been conducted as part of a collaboration between Munster Technological University and University College Dublin. And we've discovered something really important. We have been able to demonstrate that it is possible to reduce nitrogen by about 20% with no compromise on the yield and quality of the grass produced. What we've done is we've coated the fertilizer, in this case is calcium ammonium nitrate granules, with the algal biostimulant that's produced by our industry partner Brandon Bioscience. It allows us to apply less fertilizer, but also at the same time allowing us to get high yielding grass and good quality grass with less input.
So these are lysimeters. They're like mini farms. They're all contained different soil types that we've obtained from the south and southwest of Ireland. We're kind of looking at the effect of different soils and the variations that brings about on our results. We've got clover-rich, plantain-rich, chicory-rich, different types of multi-species sward. Some soils you got a bit more perennial ryegrass and so on. By having them right beside us here, beside the lab, what we can do is apply different rates of nitrogen and see the effect of the linearity of the nitrogen levels versus our treatment.
What we are going to do in this project is to look at beneficial microbes as well. So there are essentially bacteria that will fix atmospheric nitrogen and bacteria that will mobilize phosphorus as well. And what we're going to do as part of this project is to combine both the algal biostimulant and the beneficial microbes as a way to see whether we can further reduce the amount of fertilizer added onto farms. What we have demonstrated is it's possible to make farming systems more sustainable, more environmentally friendly. The reduction in the use of fertilizer translates to a reduction in the emissions from the farms. And that's a benefit not only to the farmers, but to society as a whole.
Thank you very much, Minister. We've got some presentations for you now. Again, a reminder about Slido for questions, and I'd ask all of the speakers to please stick to their allotted time. Our first presentation is from Karl Richards, and Karl is head of Teagasc Climate Centre.
Good morning, everybody, and thank you to the organisers for the invitation. So again, while I'm standing here, this is based on an awful lot of science that's happened right across Teagasc, but also the universities that we work with too. Just to remind you, why are we here? Agriculture is a really important part of our national inventory, 37.7% in 2023. And in terms of what those gases are, as mentioned, 75% of it's methane and 22% of it's nitrous oxide, which is laughing gas, as most people would know it. And we're going to deal with that primarily in this session. So within the nitrous oxide, 41% of nitrous oxide emissions come from fertiliser use, 31% comes from the dung and urine that our animals deposit during grazing, and the remaining 13% comes from manure and manure application, and that's the basis behind 3 of the talks that we're giving this morning. Another reminder again, so Teagasc we produced back in 2023 our marginal abatement cost curve, which is a menu or a suite of options, technical options for farmers to reduce emissions. And what you'll see on the graph here is that on the vertical axis it's the cost per tonne of CO2 abated. Anything below 0 is sort of a cost saving. Ultimately it should reduce costs on the farm, and a lot of these result in reductions in nitrogen fertiliser. The bars that are above the line are obviously cost positive, and the width of the bars indicates our estimation of how much those technologies might reduce emissions by. Fertiliser formulation is a key one, but the first 3 or 4 of those actually reduce nitrogen fertiliser requirement in the first place. We know when we reduce nitrogen fertiliser, so here you see the rate of nitrogen fertiliser for different types of fertiliser, and then the cumulative nitrous oxide emissions on the vertical axis. So we can see as we move from, we can't apply 500 anymore, but we did the experiment to test that out, but from 200 kilograms downwards, you can see we can linearly reduce nitrous oxide emissions. So calcium ammonium nitrate is the higher dotted line, and urea or protected urea or urea with the urease inhibitor are the lower line. So irrespective of the fertiliser you use, if you reduce the amount of fertiliser, you reduce your emissions. You also reduce costs on your farm as well. Why are we interested in nitrogen and where do these emissions come from? This is a basic nitrogen cycle. Plants uptake the ammonium, the NH4, or the nitrate, the NO3 forms of nitrogen in the soil. They can't uptake organic fractions. They can't really take them up. So what we're doing in terms of inhibitors is we're trying to stop 2 pathways. The first one is the formation of ammonium, and that's using a urease inhibitor or protected urea. And the other pathway we can block as well is the oxidation of ammonium to nitrate, and that's a nitrification inhibitor. They both work in different parts of the nitrogen cycle. What they do is reduce nitrate formation in the soil, and nitrate is the source of nitrous oxide primarily in our wet soils. It's also a source of nitrate leaching. So a lot of science we've done over the years has shown fertiliser type is really important. So the top graph here shows calcium ammonium nitrate, our dominant fertiliser back in 2018, and that has emissions of about 1.5%. But we can see the urea with MBPT, which is protected urea, dramatically reduce those emissions by between 70 and 80%. But when we start to introduce a nitrification inhibitor, so that's the second part of the pathway that I showed you, we get inhibition above 80%, and actually statistically no difference between 200 kilos of fertiliser or where we applied 0 fertiliser. So really, really effective at reducing emissions. At the same time, we also need to be conscious of ammonia emissions, because while it's an indirect greenhouse gas, it's an atmospheric pollutant. And we can see with our protected urea, when we use those, we reduce ammonia emissions by about 75% compared to straight urea. We also note there, when we use a nitrification inhibitor with just urea, you can actually end up increasing emissions. So, that's why it's important to have the 2 inhibitors. Some recent research that we've literally just published in the last month from my colleague Patrick Forrestal is looking again at further nitrification inhibitors, and these are ones that are commercially available. So the experiment he's done here is in two seasons, spring and autumn, and you can see the urea on the left. And when we use nitrification inhibitors here, we reduce the nitrous oxide emissions by between 12 and nearly 70%. So those are the 4 different nitrification inhibitors. You also see they're all different. So the type of nitrification inhibitor or the type of urease inhibitor has different efficacies at reducing emissions. And it's important that we capture that and that we can apply that in our national inventory. All diesel cars don't have the same fuel economy, or all cars don't have the same kilowatts per 100 kilometres. So it's really important that we we quantify and use the correct efficiency factors for our fertilisers. But here again, it indicates if we change our fertiliser type, we can further reduce emissions. But there are concerns. So there are concerns around the safety of different fertilisers, and there's an awful lot of legislation to get your fertiliser approved. But here's some of the research that we did after we did the protected urea research on nitrous oxide. We had to look at, is there going to be an effect on food safety? So again, a lot of this had been done during the registration of the fertiliser, but we had to repeat it to ensure that there was no risk to the food that we produce or to the environment that we farm as well, so we can't negatively impact on the environment. We developed some really new sophisticated analytical techniques to get down to really, really low levels. You're talking a drop of blood in a swimming pool type levels of parts per billion or parts per trillion. So really, really low levels. And I suppose the end of the work and the risk assessment modelling that we did as part of this research, which was done in UCD, concluded that there was no risk of either the urease or the nitrification inhibitor that we use, DCD. They did not present a significant risk to human health. We've got new techniques and new tools that we can use to support our research. Here, this is an automated chamber method that we can use to measure nitrous oxide emissions and methane and CO2. It measures it every 15 minutes, every day, for however long you switch them on. And this is a fertiliser event that my student applied, Angela, who's in the audience. So you can see here between March 27th and the 10th of April, you had an emissions peak, and after that, there was nothing. So this really helps us to better quantify the effect that our different fertilisers have on emissions. And this was funded by Research Ireland through one of their infrastructure grants. But technologies can take time. The top of this image here is, I suppose, the timeline of the protected urea story. An awful lot of research by the companies involved in developing the particular urease inhibitor from the 1990s into the mid to late 2000s. And then we did our research in the mid-2012 to 2016 period. We then did our residue research, and now we're doing more new product research. So it takes time for the research to develop, but it also takes time for the adoption of these measures to occur. So we constantly highlight, yes, knowledge transfer is important, but policies, regulations, and incentives are also really important. At the bottom figure here, we see from 2018 to 2024, our dominant nitrogen fertilisers, calcium ammonium nitrate, which is the highest emitting fertiliser. And we can see 2021 Climate Action Plan was implemented, and we can see a significant reduction year on year on the use of calcium ammonium nitrate. It's down to just above 50% now. Our target is to reduce that to less than 10%. So we're getting there. We can see protected urea has increased from 0 in 2018 up to close to 30% in 2024. But again, time is ticking, and we need to expedite these, the adoption of these technologies that we know work. There are new areas emerging that research is working on. These include very low nitrogen use or no nitrogen fertilizer systems, looking at the impact that soil fertility has on the actual emissions themselves and the soil biology, because these emissions are primarily driven by the soil biology. Looking at different types of fertilizers, liquid and bio-based, looking at new inhibitor technologies, looking at biostimulants, as the previous speaker in the video, Karl, mentioned, the work in UCD. Precision fertilizer management, but also, as highlighted this morning by Conor Murphy, we're not living in a static climate. The climate is changing, so we also need to make sure that the measures we're introducing are climate-proofed and that they can, I suppose, sustain the changing climate that we're going to see in the next 10 or 20 years' time. We've new technologies to measure the emissions, and that makes the research, I suppose, easier and reduces the uncertainty around the measurements that we make. But there is the whole area around perception and acceptability of our measures. So with that, I want to thank you very much for your attention. I want to thank my colleagues who contributed work to this across the island of Ireland, and the funders who funded this research. Thank you.
Thank you very much, Karl. Our next speaker is Dominika Krol, who's a research officer with Teagasc.
Thank you very much. Welcome, everyone. Thank you so much for the opportunity to speak with you today. So I'll jump right in, and as Karl already set the scene about the importance of agricultural nitrous oxide emissions, I'll focus my talk on manure management and grazing excreta. So we've heard that approximately a fifth of agricultural GHGs come from nitrous oxide, and within that, approximately 40% comes from management of animal excreta, be it urine and dung deposited at pasture by grazing animals or by application of animal manures to soils. So this category is actually just as large as fertiliser source, synthetic fertiliser source. So it's quite sizable. And the question is, I kind of want to explain why and how to quantify and mitigate those emissions. So first of all, why? Animals, whenever they ingest the nitrogen in their feed, they are not very efficient at converting it into their products such as milk and meat. So between 70 up to 95% of nitrogen they ingest is excreted. And if it's excreted at pasture, it ends up in a very small patches of concentrated nitrogen loading, way in excess of grass demand. So what happens is that nitrogen can then give rise to emissions, be it nitrous oxide, be it air pollutant ammonia, or through leaching into the ground. So first of all, the question is to quantify those emissions accurately so that we know how to reduce them. So a few years ago, we had a joint study between Teagasc and AFBI where we applied urine and dung patches to pastures in spring, summer and autumn on 3 different soil types in Ireland. And here what you see on this graph is percentage of nitrous oxide emissions after application. What I want you to take from this graph is that emissions from dung are very low, emissions from urine are typically higher, but most of them are below the IPCC guideline that we still use. However, in a perfect storm environment, when you apply urine to heavy soils in autumn, you can end up with very high emissions, and that really drives the importance of grazing management and reduce kind of management impacts at the shoulder of the year. But then the question further of, well, how can we mitigate those emissions? So in an ongoing study between kind of in Teagasc and colleagues with Teagasc Moor Park, we've fed animals 3 different diets. So they were grazing grass only, grass clover and grass clover plantain swards. And what we've noticed here, the graph on the left-hand side, is that cows produced more urine when plantain was included in grass sward. At the same time, in the middle graph, you see that concentration of nitrogen in that urine decreased. So overall, putting those 2 metrics together, the nitrogen excretion was lower from the pasture that contained clover, and then even lower when we introduced plantain as much. So inclusion of clover and plantain can reduce urinary nitrogen in cows, and that can in turn reduce emissions. So how's that? Here again, an ongoing study from a PhD student of mine, Ernest, who's in the audience here. Ernest applied urine patches to grassland swards at different concentrations, and you can see that with decreasing concentration of urine in the patch, of urine nitrogen in the urine patch, we can decrease emissions of both ammonia on the left-hand side and nitrous oxide on the right-hand side. But there's even more to it, because he applied those patches to 3 different sward types. So grass only, grass clover, and here we had multi-species, so that contained grass clover, plantain, and also chicory. And lowering nitrogen concentration reduced emissions, but because we applied it to different pastures, we also saw something else. The increase of emissions with increasing urine loading is different between those pastures. So it's the highest for grass only, lower for grass clover, and the lowest for multi-species. So this is really something to think about in terms of how animal diet can impact emissions associated with grazing. And here, just want to draw your attention to kind of how moving on from a kind of a research situation to maybe more of a real live grazing scenario where we can identify and mitigate patches. So in the past, we've used drone technologies and also proximal sensing technologies to spot urine patches within pasture. We can also use advanced cutting-edge methods to measure emissions from those patches at a paddock scale. But ultimately what it leads to is technologies such as what's available in New Zealand now, which is a robot that drives over pasture to identify urine patches, deliver nitrification inhibitor right into them to reduce emissions, and also is able to apply variable rate of nitrogen fertilizer so as to not over-fertilize patches. So that kind of gives you a flavour of reducing emissions both by kind of by grazing management, by what type of swards and diet our cows ingest, but also kind of technical solutions such as this. So there's plenty of really, positive and exciting opportunities to reduce emissions. So I'll have to move on now to the latter part of manure management, which is a manure application to soils. So here, what you see here is, so in terms of manures, I'll focus on slurry, which is liquid manure, because that's what mostly we operate with in Ireland. And you're probably familiar with some of the mitigation measures, how to reduce emissions. They are mostly aimed at reducing ammonia, which is quite a large loss from a manure source. But the question is, what impact does it have on nitrous oxide? Well, if you look at the graph on the left, and we look here at impact of timing of application of slurry, there's not an easy answer, but we do say for ammonia emissions, if we move towards spring application, we can reduce emissions because the summer applications might be the highest. And here also we saw the highest emissions for nitrous oxide emissions. In terms of methods of application, low emission slurry spreading that you can see represented on the right show that there is actually no discernible increase from using those methods on nitrous oxide emissions, except for a little increase for injection, which is not really a popular method in Ireland. And that just kind of shows us that those mitigation methods that we use for mitigating ammonia don't have negative impacts, and they can have positive impacts as well on mitigating nitrous oxide. When we look further, other mitigation methods for manure spreading is things like additives and inhibitors. So on the left-hand side, you see acidification added on top of the application method. Acidification, so adding acid to slurry to reduce its pH in order to reduce ammonia emissions, is a very effective way of mitigating those losses, but it doesn't have negative impact on nitrous oxide emissions, so it can be confidently used without exacerbating those. Similarly, on the right-hand side, the different inhibitors and additives that we're testing at the moment, they are not increasing nitrous oxide emissions. So they can be used confidently without potential problems. So that was a pit stop tour of what we've done and what we're doing so far, but there's also emerging areas of where we want to focus our efforts in the next while. So obviously, as I mentioned, animal diet is important, and there's animal feed and additives coming on stream at the moment. We don't know that much about emissions from the excreta following animal ingestion, so that's a line of work that we're exploring at the moment. Also, anaerobic digestion of agricultural feedstocks is becoming more popular, and the question is how those digestates perform as fertilizers. And then also various slurry treatments, be it physical, chemical, such as separation and novel additives, recovery of nutrients from manures, such as through renure, and as Karl mentioned, using new techniques to measure emissions that gives us much more opportunities at better quantification of emissions. And here, just to give you a flavour, there's a graph of our comparison of emissions from digestate versus calcium ammonium nitrate applied at 2 fertiliser rate, and at both rates digestate actually halved emissions from CAN. So on that note, I just want to acknowledge my co-authors, without whom all those work wouldn't have been possible. Thank you to colleagues within Teagasc for their technical support, and various funding bodies for funding support so that all this work was possible. Thank you so much.
Thanks, Dominika. You're staying with us. And now I'd like to invite Bridget Lynch, and Bridget is Senior Research Officer with Teagasc, to make her presentation.
Okay, thank you, Helen. To finish up the session, I'm going to focus on biological nitrogen fixation to reduce our chemical fertiliser emissions, and also biological nitrification inhibition to reduce the impact of our grazing systems on urine and dung grazing emissions, N2O emissions. So starting with fixation, this is dairy systems work that was led by Brendan Horan in Moore Park, that's just been recently published, where he has investigated over the last number of years perennial ryegrass receiving 250 kilos of chemical nitrogen compared to grass clover and a multi-species system receiving half the chemical fertiliser at 125 kilos of chemical nitrogen per hectare per year. In the study, they have shown an increase in annual milk per cow per year and milk solids yield, moving from the grass only to a grass clover system, and then a further increase in yield as we diversified the swards further to the multi-species swards. This increase in yield was achieved in the spring and summer period, as you can see in the lactation graph on the bottom, and there was no major impact on annual herbage yield across treatments. They were quite similar. So this fixed nitrogen is obviously going to reduce the fertiliser emissions. In Johnstown Castle, I suppose we've been doing similar work, but at lower levels of chemical fertiliser. So we've been comparing ryegrass dominant clover system that's getting, over the first 3 years, an average of 178 kilos of chemical nitrogen. And we compared that to a multi-species getting 67 kilos of chemical nitrogen over the first 3 years of the study. And we've seen no difference between our treatments with regards to animal production or in herbage yield during the first 3 years. So we've been able to almost half that chemical nitrogen that we saw in the initial study in Moore Park in what we're doing in Johnstown, and still, I suppose, retaining good animal production and herbage production on those grazing systems. So moving on to inhibitors, and Karl covered chemical inhibitors in his study. I suppose they are effective. We did quite a bit of work on DCD back in the day. This is work that PJ O'Connor did, where he pulse dosed dairy cows with DCD and applied their urine to lysimeters and to grass plots as well, to look at nitrous oxide emissions and nitrate leaching and herbage production. You can see from the graph, the orange bar is the urine plus DCD, and the green bar in the middle is the urine only, and we had quite large reductions in nitrate leaching and also in nitrous oxide emissions from the chemical inhibitors. So in addition to the ongoing research that's been done on the chemical inhibitors that Karl covered, there's also a focus on biological nitrification inhibition. So what is BNI? So it's a nature-based solution and a mitigation strategy. So we can select sward plants that will grow in our grasslands that the cows can consume. There's BNI compounds in the leaves of these plants that are active when the animals consume them, but they're also present in the root exodus. So in the root exodus, the BNI compounds are shed, and they have an impact on the nitrifier bacteria. So they prevent the conversion of ammonium to nitrate, and obviously that nitrate can be lost through nitrate leaching and also in the correct conditions through nitrous oxide emissions. So a new review paper that has come out from MPI's John Roach in the Journal of Dairy Science has shown that the reduction from plantain-containing swards in urinary nitrogen excretion is about 7% for every 10% increase in plantain. So just looking at plant species, and this is a nice study that our project partners in AgResearch did, led by Suzanne, where they looked at a number of plant species, applied common urine onto lysimeters and measured nitrous oxide emissions. As you can see, there's quite a wide range in the nitrous oxide emitted per hectare from those plant species. And towards the bottom of the scale, you can see that there is plantain and there's different cultivars of perennial ryegrass listed there as well. So in our research programme, we're focusing on perennial ryegrass and on plantain as well. And these are plant species that we grow very well in Ireland. So from a multi-species sward point of view, and we've had published work that has shown a reduction in nitrous oxide emissions and in yield scaled intensity. So when you express nitrous oxide on a yield point of view, we've seen reductions in nitrous oxide emissions, and this builds on Dominika's work. Plantain is the key species here in the multi-species sward. So if you look at the graph, or on the graph on the slide, you can see that there's lower emissions towards the herb part of the sward. So plantain-containing swards reduce soil nitrate through nitrification inhibition. And I suppose this is a tool, it's a manipulation of the grassland composition that is a very practical and scalable farm level solution. So building on that work that we've done on the multi-species sward, we've been focusing on simpler mixes and seeing what we can do with plantain. So this is work that my colleague Patrick Forrestal has recently published, looking at grass clover plus 30% plantain, and then grass clover plus 50% plantain. And in this study, it's been looking at nitrate leaching. And we saw a reduction across a 2-year period in nitrate leaching from both the 30% and the 50% plantain treatments of 58 and 71% respectively in nitrate per hectare per year. So I suppose it's shown the co-benefit of the likes of plantain and BNI to not only reduce nitrous oxide, but also to reduce nitrate leaching. So the emerging work and the ongoing work that we have, so we have, through the benefit of the Ireland-New Zealand Joint Research Initiative, have a project called BNI Pastures with project partners AgResearch, UCC and Trinity, where we're building on the work, I suppose, that I have presented, where we look at the soil sward mechanism and the work that Dominika has shown on feeding the dairy cows, where we're going to combine those and apply urine from cows that are grazing plantain-containing swards and apply that to swards themselves and look at both mechanisms and see what reductions we can potentially get in both nitrous oxide and nitrate leaching as well. William Burchell and his student Peter Harper are focusing on urine patches, so urine frequency, concentration, and urine patch size from dairy cows that are grazing plantain-containing swards. And they're also looking at urine patch size and concentration and looking at nitrous oxide emissions from those as well. So that's all work that's ongoing. And not to leave the tillage sector out, we also have a project called Vertigo, funded through the Green Area Hub, where we're looking at plantain inclusion in cover crops and legume-containing cover crops to see if we can stabilise that fixed nitrogen from those cover crops over the winter period, and that may be of benefit to the following crop that's sown after the cover crop. And we're also looking at plantain inclusion in short-term lays in tillage systems where we want to see if the plantain can stabilise that fixed nitrogen in the short-term grassland lay and make more of it available in the following spring barley crop. From a perennial ryegrass point of view, I suppose the beauty of the joint initiative with New Zealand is that we were able to send a PhD student over to AgResearch, where she learned about the screening method that you see there, that's called hydroponics. That's now been set up in Teagasc Oak Park, and she is going to screen the Irish genetic perennial ryegrass resource for BNI activities, so we'll be able to see what populations have high and low BNI in our own perennial ryegrass varieties, and we will sow them into soil to see if that BNI activity carries through, and they're also going to look at gene expression as well. So really exciting work as part of that project. So in conclusion, our low nitrogen system, so I suppose across all our grazing systems, we're continuing to push that chemical nitrogen down and hold on performance and productivity. I suppose further sward diversity is starting to show, we're starting to build evidence with regards to additional benefits. BNI is a nature-based solution that has the co-benefit of also reducing nitrous oxide and nitrate leaching loss. And I suppose across our Teagasc grazing systems and in our partner universities and institutes, we're continuing to, I suppose, do a full systems appraisals, not just to look at component work. So assess the productivity, the gaseous emissions, water quality, soil health, and apply all that data into life cycle analysis. So with that, I would just like to acknowledge the contributors to my presentation and to the funders of the research that we've presented today. So thank you.
Thank you very much, Bridget. There's a lot of questions coming through on Slido. I'll put them through to the panel now. But just a reminder, if you can keep the Slido questions, make sure the questions are related to the presentations that are ongoing. It's just a better way of making sure your questions are asked. So the top question that's come through, and I think this might be for you, Dominika, that what mechanism in the multi-species sward reduces nitrous oxide emissions, like during the urine trial that you mentioned, and do different species take up or fix larger amounts of it?
So I think Bridget already answered some of that question in her talk, because she highlighted that in mixed species, plantain in particular has the biological nitrogen inhibition impact, whereby it can reduce nitrate concentration in the soil, so that reduces nitrous oxide emissions. But also plantain has a diuretic effect, so that's how cows that ingest plantain end up urinating more, but diluting that nitrogen concentration. So there's a couple of effects kind of stacking on top.
You're happy with the answer, Bridget, that Dominika just gave?
Yeah, that's exactly it.
Okay, there's another question to the panel at large. Is Teagasc investigating the behavioural science aspects of climate action, specifically how to encourage farmers to adopt these new innovations? Is that something you're researching, Karl?
Absolutely. Colleagues in our rural economy programme are doing that in association with the Signpost programme, which maybe we're hearing a bit about later, but absolutely really key.
Any idea on any findings or anything that's coming out of that research, or is it too early to tell?
Well, I think at a general high level, farmers are really willing and happy to adopt technologies, but there's a challenge around when those technologies cost them money, or if there's, I suppose, a cost to them in terms of increasing management complexity and things like that. But maybe Tom can pick that up in the afternoon.
Okay, another question. Do any of these efforts to reduce nitrous oxide from soil have any effect on soil health? Do they have any impact on it? Anyone like to answer that?
Yeah, on the chemical inhibitor side of stuff, we did some work on that on our long-term experimental trials, and we found no significant effect on soil biology specifically. And I think maybe, Bridget, you might want to say something on the multi-species soil health.
Yeah, so our colleagues in Johnstown, Fiona Brennan's group, along with John Finn, have been looking at soil health indicators from multi-species swards, and they have shown an increase in soil nematode populations and diversity of those under multi-species swards. So very positive indicators on the soil health point.
So positive impact then. And it's something, Karl, you mentioned in your presentation about the impact on human health, no significant impact on human health. Does that mean a little bit of an impact on human health?
There was no impact, and even more importantly, we now have a risk assessment model that we can plug other chemicals into and assess the effect that they might have on human health. So really positive, and really important to try and get, I suppose, science-based residue limits into food safety legislation and things like that,
Okay.
and codex. So I think really important.
Okay, it's a good way of getting, I suppose, consumer and the wider public buy-in then as well, isn't it?
It can't have an impact on food safety or environmental safety there.
And the message needs to get out as well very clearly about that. Okay, a question from Tallav Bio. What lessons can be learned or strategies borrowed, if any, from organic farming in tackling soil nitrous oxide emissions? So is there anything we can pick up from organic farming? Anyone like to take that?
I think probably the use of alternative pasture species. So in organic farming, they haven't been able to use chemical fertilizers. So they have been using, clover, red clover, multi-species swards and very diverse swards in order to grow their herbage to feed their animals. So I suppose we can learn from their work and from their experience in a no chemical environment, the impact and the potential for reducing N2O emissions.
Okay, Dominika, have you anything to add to that from organic farming?
I think Bridget covered that,
Okay.
except that, we should be talking to organic farmers because they have so much more experience, maybe even than researchers in this area. They've been doing this on the ground.
Another way of knowledge transfer then really, isn't it, from the organic sector?
Exactly.
Okay, TJ Flanagan, what will be the implications of failure to get biomethane up and running at scale?
So maybe I'm not going to deal with failure, but biomethane and biomethane strategy is a really important part of the marginal abatement cost curve. One, it replaces chemical fertiliser by producing a really good, high quality, highly available nitrogen fertiliser source, and the other is around diversification options for farmers, that they're able to maybe focus on producing biomass to go into those stores, but it also reduces methane from manure storage as well. So it's a really important measure, and it's important that we see it expedited.
Speaking of expediting things, a question from Dave Fitzgerald. How do we accelerate the research and adoption timeline? Is there work ongoing in this? Is that just about funding and more people in, scientists? What's it about?
I think no.
Dominika,
it's not just research. We have very good research and very good research outputs that we then pass on to, within Teagasc anyway, to our advisory, specialist advisors, who then brief their advisors, and that's how the message spreads out. And then via our signpost farms and via farm walks and those disseminations where farmers can adopt new measures and learn from each other. I think there's, it has to be a concerted effort of not just research, but then putting that research into action.
And it reminds me of sort of what was said during COVID, that perfection sometimes can be the enemy of success and can slow things down. And because, I suppose, time is of the essence, we don't need to achieve perfection.
No, the science is really clear on those technologies in the market, that they work and that they significantly reduce emissions. The example I always give is low emission slurry spreading. We did the science in the early 2000s. We knew it reduces ammonia emissions. We knew it was really important. Knowledge transfer tried to transfer that. Adoption was very low. We then had a regulation introduced and we had an incentive introduced, and now we're at 70% adoption of that measure. So it's really clear we need knowledge transfer, we need policies and regulations, and we need incentives. Some of these technologies are really expensive to introduce, and therefore they need support. Farmers need support if they're not going to see any, I suppose, productivity benefit to that particular technology.
Okay. A question possibly from a farmer. How do you manage weeds like docks with multi-species? And this person has used multi-species very happily in some fields, but docks are a massive problem in other fields with multi-species. Bridget, you're smiling.
Yes, I'd love to know the answer to that myself. Yeah, I suppose it goes back to the principles. If you're going to sow multi-species, to make sure it's a clean sward before you sow those. So if the field already has a dock problem, I wouldn't be putting multi-species into it, because we don't have herbicides that we can apply to multi-species swards, because they'll kill the herbs as well. I think getting a good establishment and putting in a high seeding rate prevents the sward from being open when it establishes. And then I suppose when you're grazing them, my experience is that when it's wet or if they're heavy soil types, the animals will cut up those swards probably more readily than a perennial ryegrass only sward. So to try and avoid, I suppose, poaching those swards and animal treading, that would open it up for dock seeds to get in.
Okay, if we go back to organic farming, another question has come in. How are organic farms performing in terms of emissions compared to conventional grazing systems?
Really good question, and it's one that our National Farm Survey is working on at the moment in terms of getting the activity data that occur on those farms. But if you think of an organic farm, sometimes their animals are finished a bit later than a conventional system, so there's potential to maybe increase methane, but you wipe out your nitrous oxide coming from fertilizer. So the research is underway, and we will see how that pans out.
Okay, we're kind of running out of time, but maybe time for this last question. That research at Queen's University shows cattle that graze on willow leads to lower methane emissions. Is planting of willow for dairy cows to graze on selectively, could that be a solution, or is that something that's being researched here?
Maybe that's a question for the methane session.
Okay. Did you want to say something, Dominika?
Yeah, so I just wanted to add that I think in terms of willow is the tannins that are that the animals ingest when they graze on leafy and kind of plants, hedges and
so that could
trees.
be a problem.
So no, that actually the tannins reduce
They're
methane.
good. Okay.
So that's why it might be a solution. Yes, there's work ongoing, in that direction.
Okay. I think I'll throw one last one out to you. The estimated proportion of grass reseeded annually, if we get a sense of how relevant this all is. Any idea?
It's about 2% of our,
Around
about 2% is reseeded annually. But the department has incentive schemes for, multi-species and red clover swards. So there is, measures and grant aid there for farmers to put in swards. But,
okay.
If it's a practical solution, that's going to not impact on productivity, I think farmers would readily take that rather than some of the other drastic alternatives.
Okay. That's great. Well, thank you very much to our speakers, Karl Richards, Dominika, Krol and Bridget Lynch. Thank you very much, and thank you all for submitting questions. Keep the questions coming. Thank you all for your participation. We're going to move on now to session 5, which is a science and technology update where we're focusing on methane. So I'll ask the speakers to join me on the stage while we play this short film for you.
The global population is going to reach over 10 billion by 2100. So we're estimating that food production will have to increase by around 50% by 2050. Meat and milk products are going to be increasing every year to meet that demand. Ruminants are critically important from a food security perspective. They're the backbone of our thriving agri-food sector here in Ireland. However, that is set against the challenge to reduce greenhouse gases, and an important greenhouse gas in that perspective is methane. We have an international project called Rumen Gateway. It's led from Queen's University, Belfast. And here at the University of Galway, we're the Republic of Ireland lead on the project. It's a Global Research Alliance flagship project, and we're delighted that is funded nationally here by the Department of Agriculture, Food and the Marine. This is rumen fluid from an animal that was fed in a grazing system. This is the contents of the rumen of the animal where basically feed is digested and where methane is produced. The rumen is an anaerobic environment, so we mimic the conditions of the rumen and bring that inside the laboratory. This really is where science meets farming. Essentially, we're taking samples from a beef or dairy animal and then studying the microbiomes that exist in that rumen. So we can come in here to the cabinet and we can work as if we're working inside a rumen by the addition of gases that are specific to the rumen environment, that we can actually maintain that correct anaerobic environment that the microbes are used to while they're in the animal. It's really critically important to understand that the rumen microbiome plays a key role in nutrient digestion from feed and also in methane production. So these are two key issues in agriculture. So by understanding the rumen microbiome, we can then understand the mechanisms by which they act and function at a greater, deeper level. We can potentially interrupt that process to generate new biotechnological innovations that can allow us to potentially enhance digestion of feed in the rumen and potentially mitigate methane emissions. Also, this information will contribute to the design of vaccines in the future. So potentially animals, young ruminants could be vaccinated to reduce methane emissions by understanding the genome of particular methanogens that exist in the rumen.
Okay, there you have it. Our first presentation now is by Sharon Huws, and Sharon is Professor in Animal Science and Microbiology at Queen's University Belfast.
So thank you very much. I'm delighted to be invited to talk to you today a little bit about the rumen gateway. And as Helen said, my name is Sharon Huws. I'm actually the interim director for the Institute for Global Food Security, and I'm only 2 hours up the road in Queen's University Belfast, for those who don't know. So, the first talk on methane obviously has to show you just a little bit about what happens in the rumen, which is the main reason for the methane emissions from our ruminants. So methane production is perfectly normal. It's the way that the microbes that live in that rumen digest plant material. And when they digest the plant material, they produce these volatile fatty acids. And these volatile fatty acids are really useful. They're what the animal needs. It provides energy for the animal. And certain volatile fatty acids will use quite a lot of hydrogen in their production. For example, propionate utilizes hydrogen. So we really like the production of propionate because it takes hydrogen away from the other side of the equation, which is methanogenesis. The more free hydrogen you have available, the more that you're giving those methanogens in the rumen to work with in terms of producing methane. And this is the complexity that we have in the rumen. The rumen is one of the most complex microbiomes that you will find in nature. There are very few microbial ecosystems that are this complex because we have bacteria, we have protozoa, we have fungi, the methanogens and the phage all working together. And with this complexity brings quite a lot of challenges when ultimately what we're trying to do with our methane mitigation strategies is manipulate the processes in the rumen. The diagram that I have here is from the Hungate project, which was a past GRA flagship project, which really looked at what diversity of microbes we have in pure culture in the laboratories. The maybe if I try the pointer, yeah, the black marks there on these graphs show you the biodiversity that we have in the rumen. So each one of these points shows you that we have microbes here, here, and here, and we know that they all exist in the rumen. The Hungate project then asked people to isolate pure cultures, and the dots that we have here are where they managed to isolate pure cultures. This side is for bacteria, this side is for the methanogens responsible for methane production. And what the purpose of showing you this, particularly on the archaea side, you can see that we have huge gaps. And if we do not have these microbes available in pure culture, it means that we cannot understand them. So many of you would think, okay, if we fill in these gaps, that is very fundamental. How does this really translate to real agricultural practices? So if we want to put a feed additive, for example, on the market, then we need to know exactly what the mode of action for that feed additive is. At the minute, with the gaps in our knowledge on the rumen microbiome, that is challenging. So the Rumen Gateway project is really going to help us to fill some of those gaps. And the other area that I want to highlight is the work on producing a methane vaccine. You saw the gaps that we have in our methanogen cultures, and that's a huge issue because we cannot really develop methane vaccines unless we fill in those gaps in a better way. So this is what the Rumen Gateway project is about. It is about filling in those gaps, culturing more of those isolates so that we can really take forward some of the feed additives, the methane vaccines, etc. So this is a huge project. I'll show you shortly how many partners are involved across the world. But we are essentially using cutting-edge technologies to isolate some of these microbes. We are then genotyping them, so we're getting their genomes, and we're also looking at their phenotypes in terms of their ability to redirect hydrogen, because there is also the possibility that we could feed these to ruminants in order to direct that hydrogen more towards energy and productivity for the animal, as opposed to wasting energy producing methane. So it is a huge, huge project. We have 17 countries involved in this project across the world, and we're adding to it at the minute. It is funded by the Global Methane Hub, the Foundation for Food and Agriculture Research, DEFRA, the Joint Genome Institute, and DAFM here have also put quite a lot of money towards it. The reason why we've made this global is because our ruminants across the world will be consuming different diets. From the global south, they might be higher tannin-containing ones, and they might have a slight different diversity in that rumen. And our aim is to get as much diversity as possible into our culture collections. And we are very grateful for DAFM, obviously, who have partially funded this work. And they have funded Gizem, who's in the audience here as a PhD student under the supervision of Sinead and Vincent. So the progress to date, we have isolated 726 bacteria at the minute. Of those, 100 of them are novel, so they're ones where we had previous gaps. And our Chinese partner has also just updated us that they have added a further 837 novel bacteria to that mix, and many of them will be propionate producers. So we're really moving on at a pace in terms of filling those gaps. So currently what we are doing is genome sequencing them. The Chinese partner has already genome sequenced the 837. We have 100 that have gone to JGI for sequencing at the minute. And we're also, as I said, phenotyping them, really understanding their characteristics and how they could play a role in methane mitigation. And the work on isolating the methanogens has just started as well. So really quite exciting, given that we have only really launched the project in the last year, and it's quite a complicated project due to its scale. So at present, this is one of the largest Global Research Alliance flagship projects involving numerous countries that we're adding to the list. We have already isolated 950 novel bacteria that are genome sequenced, and we're looking at their phenotypes. And the interesting bit is, of course, when we get some of those methanogens coming through as well, because they will really help the development of a vaccine. We have also created an identification database to make life easy for scientists across the world. And that's the MALDI-TOF database, which we are actually going to release openly to the community soon. These microbes that we isolate as well will be open to the community. They're going to be deposited in open access culture collections, and the genomes are going to be made available to everyone, so that the ability to create impact from this in terms of methane mitigation as we move forward is as optimal as possible. And our aim really is to publish and release all of these data sets by the end of 2027. So I'm just going to finish and thank all of the funders and thank all of the partners. You can see the scale of the project from all the partners here. Thank you.
Thank you very much, Sharon. Our next speaker is Suzanne Rowe, and Suzanne Rowe is Principal Scientist with AgResearch New Zealand.
Thanks, everyone. It's my privilege to give an update on genetics and where we are with the application of genetics as a mitigation tool for methane. I'm not going to give you any facts and figures because you've had enough, so I'm going to move on. Why genetics? Well, we know that ruminant livestock evolved. They evolved a beautiful system over 40 millennia to basically harbour this beautiful, optimal microbiome within their gut and to digest C4 grasses, so indigestible grasses, across the Great Plains and convert them to energy, to meat and to milk. So we know that it's a system that's evolved, it's underpinned by genetics. What we didn't know is, was it fixed and could it be changed? And what might be the consequences of that change? So what we've discovered over the last few decades, having measured thousands of animals from Scandinavia right down to New Zealand, is that in actual fact, there's a continuum and there is variation in every single flock and herd that we look in. So it's there for the taking. There is genetic variation in the fermentation that happens in the animal and the resulting amount of methane in every system that we see. So we don't need to change breeds, we don't need to change animals into different environments. In actual fact, all we need to do is measure our own flocks and herds and pick the low methane animals because they're grazing side by side and we can't pick them out. And this is a graph of 10,000 animals that had breeding values across New Zealand. So the green bars are sheep and the blue distribution is a slide stolen shamelessly from Donna Berry on beef in Ireland. So in order to understand the implications of this, we really need to measure those individual differences. And when we started, we had very complex infrastructure in order to measure methane. Chambers that were very expensive and obviously not pragmatic or useful for on-farm measures. So the last 2 decades have really been about developing technologies that we can use at pasture. We really needed to understand what was going on in a grazing system, particularly for countries like Uruguay, Ireland and New Zealand, where it's all about that pastoral agriculture. And there are several technologies that have sprung up. So the animals with the tanks on their back, that's a dairy system in Australia, where they use tracer gases to detect methane from grazing cattle. The box with the animal's head in, that's actually green feed, and that's used across the world, and that's just where an attractant like feed is used, and there's a little sniffer in there, and it's catching, every time an animal visits it, it's catching a snapshot of how much methane that animal is breathing out. And in New Zealand, we've actually gone a little bit further and started to develop on-farm technologies that we can get a direct measure of methane. So these are simply boxes. We can pick them up with a tractor and loader. They're not attached to anything. So they're sort of a pseudo-respiration chamber, and we shut the animal in for 45 minutes. All the methane accumulates, and in that 45-minute period, we see sufficient differences between animals to be able to rank them in order to give them a lifetime ranking. So we can now rank those animals, and we know which animals on farm are low emitters and which are high emitters. So we now know a bit more about the low methane animal, having measured these animals and having measured traits alongside them. We know that they have different microbiomes. So we know there's a natural variation in microbiomes across stock, and we know that this is controlled genetically. We know they have a different energy profile, so our low emitters tend to have more propionate, and that tends to give us a little bit more lean growth. We tend to see better carcass yields from these animals, a bit less fat, and in sheep more wool. They tend to eat a bit more, and they tend to eat more frequently. But crucially, in these systems, we're seeing 4 to 20% less methane per kilo of feed eaten. So if 10 animals sit there eating the same amount of feed, we're going to get a variation amongst them in terms of the amount of methane that they emit. But most importantly of all, these animals are healthy and profitable. There's nothing to tell us that there's no reason why we shouldn't take them, breed them, select them. And this is already out in the field. So again, shamelessly stolen from Donna. These are methane breeding values for beef animals in Ireland. There's an animal here that's 10% below already that could be selected should somebody decide to breed from him. And in New Zealand, we have a demonstration flock. The National Evaluation Scheme for Methane in Sheep was rolled out in around 2018, but in 2017, on this research flock, we decided to implement genomic selection for methane. We decided to select and put it into our profit index. And what we saw was an immediate drop in methane, but crucially, no real change in our profitability. So the dotted line is the flock, and the green line is the average flock in New Zealand. And when we thought about this rolled out to 2050 and looked at that trajectory, what we saw was if this flock that we'd measured every year for 8 years stayed on the same trajectory, that we would hit target with genetics alone. And up until that point, we didn't believe that. We thought the genetics had to be combined with something else. But in actual fact, we were 40% above in terms of profitability of the average flock, and we were still going to hit that target. We should still hit that target using genetics. So I really hope that I'm here in 20 years' time telling you that. So breeding is a national solution. It's slow. It takes 2 generations to get going, and that's been a real drag factor for us, and something that's made us pause when we think about using genetics. But just like any compound interest system, as soon as it gets going, it ramps up incredibly quickly. It works in grazing animals, and it's socially acceptable. We're not doing anything new. We don't need to buy anything. We don't need to feed anything. We're just selecting what's there on the ground. The systems are in place. We don't need to set up breeding schemes because we're really good at it already, and they're already in place. We need around 2,000 to 5,000 measures a year, and that's been our key target, is measurement, managing to capture these animals and know which ones are the winners. And that one-off expense is an expense. It is difficult, but it does last generations. But it is really low cost. So in the sheep scheme, we're looking at about €1, so $2 New Zealand per tonne of CO2 equivalent saved. And if the sheep industry in New Zealand saves 0.5% to 1% a year, we're looking at 4.5 kilotons before 2040 saved, on a really, really low margin and an internal rate of return that's far superior to everything we've seen before. No one's going to get rich on it, but it's a really low impact, easy way to mitigate emissions. But obviously, as scientists, we want to go further, and I really want to plug the bilateral projects that we have. So we have a project, Methane Predict, with Teagasc and AgResearch, and what we've been thinking about is, okay, so if we have this methane signature that flows from the rumen through into milk and muscle, why can't we use the spectral profiling that we commonly use for milk composition, for herd testing, for meat quality in plant? Why can't we use this to give us a methane signature? And can we do that without touching the animal at all? So can we get signatures in our day-to-day processing that give us information on our flocks and herds? And how powerful would that be if we use that as a breeding technique? But finally, we can rank these animals and we can measure them as accurately as we like, but we're only leading this horse to water. So we're now in a situation in New Zealand where the sheep sector in particular knows what their methane emissions are. So now they now sit with a choice. There's always a trade-off. It doesn't matter what you breed for. We haven't bred for production since the 90s. We have a raft of traits that we select for. If we want to include methane as one of our key traits, we have to have an economic value. There has to be a weight. And in order for people to use those breeding values, we really need full farm economic analysis. And again, there's a roadmap collaboration, which is another New Zealand Island collaboration that is really looking into that, led by Donna. So in summary, there's genetic potential in all of our ruminant livestock. We haven't seen any negative effects to date. It's easily audible because we just did a DNA test. It's very low cost and we have very high consumer acceptance. The cumulative annual gain of 1% is commercially feasible. We don't see any sort of barriers to doing this. But we do need continued development of rapid measurements on pasture. We need to understand feed intake, we need to understand methane emissions, and we need those economic incentives in order to incorporate it into profitable, sustainable livestock schemes in the way we have for literally ever. So too many people to thank, but thank you all for having me.
Thank you very much, Suzanne. Now I'll invite David Yanez-Ruiz to make his presentation. David is a senior scientist with the Spanish National Research Council.
Thank you, Helen. Thank you for having me. Feed additives have been mentioned a couple of times this morning, and the research area around feed additives to reduce enteric methane emissions are going through a very dynamic and active period, but also equally challenging context. We know from numerous research that feed additives is one of the strategies that show the highest potential to reduce enteric methane emissions. But despite the intensive research conducted over the last 20 years, we only have 2 additives that can substantially reduce enteric methane emissions, and only one is approved to be used commercially. And another challenge is that for those additives to work, they have to be thoroughly mixed with the diet that the animal ingests. And that leaves grazing systems with no practical solutions to date. And I don't think I need to come to Ireland to show and to highlight how important grazing systems are across the world. So within this complex context, I'm going to present 2 things today. The first one is the Global Research Alliance flagship project on feed additives, aiming to facilitate the development of feed additives through providing technical guidelines and recommendations, and we'll see some details later. And then very briefly as well, I will use a couple of examples of ongoing research working on trying to address the challenges associated to grazing systems in Spain and here in Ireland. So if we start with the first one, the flagship project on feed additives is part of the Global Research Alliance. Started, I think it was 2.5 years ago. It's now completed. We had 60 researchers from different countries and institutions involved. We had funding from the Global Dairy Platform and also facilitation from the New Zealand Agricultural Greenhouse Gas Research Centre. And the outcome is basically a set of guidelines and recommendations that have been recently published as a special issue in journal Dairy Science. And the special issue contains like 6 chapters, 6 papers, and every paper addressing one of the key steps that a feed additive has to go through from the beginning to the end in order to be successful. And that includes like 2 phases. The first phase is the development, and then within the development we have different steps, starting from the initial identification of the molecules or products that can be used as additives, then going through testing at animal level, developing models to see what happens in the rumen and in the animal in the farm, and eventually also understanding how it works, as Sharon was saying before. So once these 4 steps are completed, then we move into the implementation, and there are 2 aspects that were also described in the guidelines. One is in relation to the regulation, registration to be authorized, and then how we can also account for the reductions in emissions achieved when these additives are used. So I'm going to go very briefly to show just a few examples of main things that were covered in these chapters. So we start with the first one, in which we're trying to identify candidates to be used as additives. We describe the 2 main approaches that are used at the moment. We can use the conventional empirical approach using information previous in the literature or in other labs to identify and select those candidates. But now we can go through also some more complex and modern mechanistic approach targeting methanogen's unique cellular process. And we use computer modeling in silico and docking studies in which we can model how target enzymes of the archaea can interact with different molecules. The technology developed in the last years is really helping to use these technologies nowadays. In any case, once the candidates are identified, they need to go through in vitro tests, and we also provide specific guidelines to conduct these in vitro tests. They're fairly simple in vitro tests, but sometimes some of the important things are largely ignored, and then that can affect later on the development of the additives. Once candidates are identified, then they need to go through in vivo tests, in animal trials, and this is what we cover in another chapter. This is quite a big chapter, quite a big paper covering many aspects that need to be considered before, during, and after the animal trial is conducted. And there are lots of details in this paper led by Alex Christopher and Peter Land in the paper. Another issue that we cover is what Sharon was saying before. We need to understand how it works. We need to know the mode of action. That's important for two reasons. One, because if we know how it works, then we can optimize the use across different feeding systems. So we know how to apply and when it's going to work and how. And secondly, we need this to go through the regulatory process for the approval. There are overall like 4 different types of mode of action the additives can use to reduce methane emission, depending on what step of the methanogenesis pathway they act from the beginning right to the end. But regardless of the mechanism of action, we need to fully describe how it works at the whole ecosystem level, but also microbial and molecular level. And this is how we can really optimize the use in practical conditions. And one thing that comes now very interesting in the last years is that once we know in detail how this works, then we can also design potential mixes or combinations of different additives with different mode of action that can work together and trying to find a kind of additive or synergistic effect. So if we have one additive that reduces 25%, another one 10%, but they work differently, can we combine them to achieve 35% or even higher? In theory it sounds easy, but it's not that easy. But this is an area of research that's really developing at the moment, and hopefully we'll have good results in the near future. The last part of the guidelines cover 2 important aspects in terms of the implementation. First is to outline the regulation and the evidence requirements that these regulations have for the applicants in order to get the authorization, and the critical role that the intended use, so the claim in terms of safety, efficacy, and quality that the applicants need to know to go through the process. So in the paper we described the regulatory landscape in 7 different regions of the world, and we compared the differences and similarities, and it's a very interesting exercise because this is a global problem, but the regulatory aspects are not that global. The other aspect is trying to account for the reductions in emissions, and we provide key points on the methodology to be used when we want to quantify the reduction in emissions at the very basic animal level, then going through the farm level, national, inventories, or supranational level. And this is important when we really want to capture the benefit of using these additives, and we provide information on the different methodologies, trade-offs, and also uncertainties. All this information is available in the Journal Dairy Science, in the special issue, also in the Global Research Alliance website. There is information about different webinars and activities, dissemination activities that we conducted recently, so you can get more information there, and also in the poster outside. The other part that I was going to mention today is the challenges associated to grazing systems. I'm going to use an example of work recently conducted in Ireland, where using bovar in grazing dairy cattle, there was that was provided at the morning and afternoon milking. There was a reduction only of 5% of methane, which is much lower than the 25–30% that we usually obtain in intensive systems. And the reason is because the free NOP provided at these 2 times is only active here, but not during the grazing period. We tried to address this problem in Spain, providing the free NOP in drinking water, and we could see that in contrast to the double or the 2 periods reduction, when it's provided in drinking water, there is a slow release of the free NOP in the rumen, and a more sustained delivery accounts for higher reduction. So 41 reduction of methane in comparison with 22–25 when it's provided twice a day. We understand that this is an experimental concept at the moment. We could control the drinking pattern of the animals, and this need to be tested further in different grazing conditions with different drinking water patterns. Another important example is the MAX project in Ireland, the methane abatement grazing system, which got lots of partners working not only on delivering additives for grazing system, but also a holistic view of combining different strategies in terms of supplementary manure management and also genetics. One example, very recent work, is the use of calcium peroxide as feed additives that has managed to reduce methane emissions in forage-fed animals between 10 and 30%, and in some cases also an increase in average delegating. So very promising result, very preliminary result of this project. And I'd like to finish just concluding that the guidelines, we think that can improve the capabilities of the different communities to really increase the number of feed additives that we have available. It's important to consider all aspects in the production chain need to be considered. There are other issues to consider, like long-term effects, and the scientific community is working on that. And in regard to the grazing systems, this is a big challenge, but science is also working on that, and hopefully we can have good results and good solutions in the near future. Thank you.
Thank you very much, David. And the final presentation of this session is by Paul Crosson, and Paul is a research officer and beef enterprise leader with Teagasc.
Okay, thank you, Helen, and good afternoon, ladies and gentlemen. So the previous slides looked at, I suppose, interventions that can be taken to reduce methane emissions. So over the next number of slides, I'm going to look at how can general management impact methane emissions on dairy and beef cattle production systems. I'm going to look at three areas: nutrition. I'm going to break that into grazing and indoor feeding. We know the importance of grazing in our Irish pasture-based system, so I'm going to look at that firstly, and then indoor feeding. Finishing age, which, as we know, particularly the Irish delegates here, really important part of our climate action plan, and age at first calving. So firstly, looking at nutrition, and from the point of view of grazing management, what impact does better grazing or improved grazing management have on methane emissions? I'm going to pitch this in the context of two different studies: a beef study carried out at Lyons and reported by Tommy Boland, and a dairy study carried out at Teagasc Moor Park. Both studies have two measurement periods, so an early measurement period and a later measurement period. And within each measurement period, we have good pasture management, you might call it, and suboptimal pasture management. And what we can see is a very general trend across both studies. And this is pretty standard in the literature in terms of the effect of grazing management. For the second measurement period in particular, for the optimal grazing management practices, we have very clearly lower levels of methane emissions per head per day. Of course, we're also really interested in the importance of animal performance. So if we look at this in the context of animal performance, from the beef system's point of view, daily gain, from the dairy system's point of view, milk solids yield, we can see very clear advantage of improved pasture management on animal performance. So putting the two together, if we look at the impact of pasture management on methane per unit of animal product, we see up to a 30% difference in methane intensity. So simple pasture management can have a really important impact on methane emissions. Of course, monitoring and verification for inventory purposes is very difficult, but the evidence is very clear in terms of its effectiveness. So lower pasture herbage mass, in effect better pasture management, higher digestibility forage, higher animal performance, lower methane per head, and much lower methane per unit of product, particularly for that second measurement period. If we move to indoor feeding, for forage-based systems, and this is from work carried out by Chan Hai Yan in Athby Hillsborough in Northern Ireland, similar results in a broad sense: higher quality forage, lower methane per unit of animal product produced. But of course, in animal systems, particularly during indoor periods, we will often have supplementation provided. So I wanted to look at a scenario where we had supplementation provided over the winter period in beef finishing systems. This is a study carried out by Padraig O'Reilly and Emma McGeough at Teagasc Grange. There were a number of different feeding treatments. I'm going to report on 4 of them here: a low-grain whole crop system, a high-grain whole crop wheat system, a standard grass silage concentrate system, and an ad libitum concentrate system. And if we think of it in terms of starch content of the different diets, you're going from 20% starch to 40% starch, the grass silage concentrate about 7% starch, and the ad libitum concentrate in the high 40% starch. And we know that starch has a really important impact on methane emissions in that it lowers methane emissions in feeding systems. And we can see that effect coming through very clearly here. Methane per head per day, much lower for the ad lib concentrate system. In terms of the forage systems, our high grain system in whole crop being next lowest, and highest for our grass silage and our low grain systems. Animal performance also very strongly affected by grain content or starch content in the diet, much higher for the ad libitum concentrate system relative to our other systems. So putting the two together in terms of methane emissions per unit of animal product, much lower for our ad libitum concentrate system relative to our other feed systems in an indoor scenario. But of course, we have other metrics. We're focusing on methane in this session, but of course there are other metrics that are really important, not least broader emissions impacts. I know there is a session on land and soil emissions later. So you have your tillage system in terms of your ad lib concentrate versus your grass-based system. You also have impacts in terms of economics. Is it economical in terms of ad libitum concentrate system? Possibly at the moment. In the long term, more questionable. And also broader sustainability criteria, food, feed, and so on. Okay, moving to finishing age. So a really important metric in the context of the Irish climate plan. Karl mentioned the Teagasc marginal abatement cost curve earlier. In the Teagasc marginal abatement cost curve, we have proposed a 3-month reduction taken from the Irish Climate Action Plan. If that were to be achieved, it would lead to an abatement of about 730 kilotonnes. So how are we performing? Probably a little bit more of a mixed message here. And that's putting it maybe kindly in terms of the performance on finishing age. If we look at it in a 10-year time frame, for steers, we've reduced finishing age by about a month. And for heifers and young bulls, there's no change. But if we look at it using our base year for our Climate Act of 2018, we have a 1-week increase for steers and about a 2-week increase for heifers. So a lot of action needed in terms of finishing age. To be fair, if we looked at it up to 2022, we were trending in the right direction. Conor Murphy earlier this morning presented some of the impact of climate change on our intense weather events. So if we look at what happened in 2023, the very wet and difficult back end of that year and the start of 2024, that largely explains a lot of the loss in progress that was made on finishing age. So those large increases really driving us back in terms of finishing age. Probably that 730 kilotonnes is getting very challenging now in terms of achieving that in our climate plan. So what can we do? If we look at some of the actions and some of the factors affecting finishing age, really important piece of work published by Natasha Munier from Animal Health Ireland, looking at the effect of animal health incidences on finishing age, up to 46 days in the case of liver fluke. So managing herd health, really important. I'll also point to a project led by Paul Smith at Teagasc Grange and funded by the Department of Agriculture here, the Beef Quest project. In that project, it's looking at factors that affect finishing age and how can we, I suppose, promote best practice to reduce finishing age. The first phase of that is, I suppose, a detailed animals or farm study to see what are the differences from farm to farm in terms of finishing age. We know animal to animal that there are very large variations, but what was really interesting is looking at the farm to farm variation. And based on predicted 400-day weights, we have the potential of a 106-day difference in finishing age between the top and bottom third. That's at farm level, not animal level. So the next stage of that project is to delve a little bit further. What are the key farm and animal level drivers affecting this? And indeed, there was a question earlier on behavioural issues. So what is driving behaviours at farm level that's underpinning this? First calving age, again, a really important metric in respect of our methane emissions. If we look at average first calving age in Ireland in 2024, suckler heifers over 31 months, so in our suckler herd, the dairy herd about 26 months. So in particular, there are opportunities here in our suckler herd to reduce finishing age. We have a very detailed database through the Irish Cattle Breeding Federation, and we can delve into some of the outcomes of different calving ages. And that shows that cows which first calved at 2 years of age have similar productive output over their lifetime compared to later calving ages. It also, I suppose, really importantly, increases production costs. For example, on suckler farms, it's about €50 for each month delay in calving age. So that's a real cost at farm level. And as I said, it's a particular opportunity, I would say, for suckler systems. I suppose the positive around it is that if we address issues around finishing age, issues around, there are challenges with certain later maturing breeds. We know there are nutritional, particularly first winter and winter performance levels that are suboptimal, that are impacting on age of puberty and finishing age in terms of the previous session or the previous slide, and health, of course. So addressing those for finishing age will also improve our ability to meet our first calving age targets. So in summary, grazing management and feed quality characteristics have a really important impact. Reducing finishing age tends to decrease emission, and I say tends to because when you get to a certain level in our blueprint systems, reducing finishing age further is unlikely to generate further savings, and in fact could move in the opposite direction. But in general, in relation to the national finishing age average, the weighted average for finishing age, reducing it will reduce methane emissions. It's showing little progress. Similarly, age at first calving, we are seeing very limited progress. We know the factors, so ongoing projects are seeking to, I suppose, uncover those practices and importantly, again, the behaviours affecting uptake, not just of emissions reducing technologies, but also those profit enhancing technologies, and those, in this case, are going hand in hand. So thank you very much.
Thank you very much, Paul. I might come to you, Sharon, first with a question. It was mentioned in the opening video at Rumen Gateway project, and you also mentioned it, the possibility of a methane vaccine. And if we're looking at that possibility, how far down the line are we looking before something like that could become available?
Well, there is a new project funded by the Global Methane Hub, which is focused on developing that methane vaccine. And perhaps David can follow up afterwards as well, because he is part of that project. But that is set up, in I believe it's a 3 or 4 year project to try and develop that. So Rumen Gateway, of course, is critical for that, because we have to have the genomes available for the methanogens so that the vaccine can then cover most of them. Because the danger is that you don't want to cover some of them and the others take their place.
Okay,
maybe David.
Yeah, very quickly. The project started like a year ago, and it's got 2 components. One, looking at how does the immune system of the animal recognizes the methanogens when they come into the digestive tract when after birth, trying to understand that those first weeks of life to see how we can knock them down, and then there's another part looking at what components of the archaea are the key parts of the cells that are recognized by the immune system. And it's important to consider this because basically what we want to do is to kind of fighting against nature. Those microbes are symbionts of the rumen. So I would say that at least 3 or 4 more years until we have results that can show something that can be
So soon enough then, really, in the grand scheme of things. And the research, Sharon, that you touched on, if it comes into practice, would it make a difference to farmers on the ground? I mean, if they are looking at methane mitigation, will it make economic sense for them as well, do you think, if this is something that can help them?
So without the Rumen Gateway project, the development of feed additives will be hindered, because we won't have the baseline knowledge to understand their mode of action. So it underpins a lot of the development of any feed additive, and those feed additives can be, economically viable in various different ways. But if we talk about maybe using them as direct-fed microbials, feeding them directly, then that is what we're talking to companies about now. How might that look like? And I know partners in AgResearch are also developing some potential direct-fed microbials. So we're at the early phases, of defining the economical status of direct-fed microbials.
Okay.
But it's
something, it's a big part of it.
Yeah.
Suzanne, you were looking at genetics. And if we do find the animal that's genetically predisposed and genetically ideal when it comes to lower methane emissions, are there trade-offs? I mean, as regards maybe meat quality, as regards... So I mean, could it be good in one aspect and the animal not so good in another aspect?
So we haven't seen any in terms of meat composition and meat yield. It all looks pretty good. Mostly it doesn't correlate with anything. It's pretty benign, which is why it's just segregating there cheerfully. The only trade-off that we really see is that if we take our foot off the gas in one direction, that we're obviously going to lose some of our productivity gains. We might go a little bit slower.
Okay. David, I suppose a lot of this is about getting the message out there. And I mean, you even touched on it, Suzanne, in your presentation, the importance of measures being socially acceptable. But a lot of that is getting the message out there. And we saw what happened recently when I suppose that Arla in the UK were promoting the 3-NOP feed additive. And all of a sudden there was a massive social media storm. We saw people pouring milk down drains. We heard talks about infertility, about cancer risks. So the claims were quite kind of outrageous. And so is it very hard to counter those claims because your message isn't as simple as those messages are sometimes?
Yeah, that was tough. I think it was quite sad and frustrating to see how a few tweets and some social media posts could completely ignore and overshadow so much science. This particular additive is approved for commercial use, and the European, in this case, the European legislation is very protective. It's taking 12 to 15 years for that additive to get through all the steps of evaluation. 28 independent scientific scientists in the panel. So the first thing is that it's important to put the science in front and show the evidence. Also, it's very important to pass the right message at the right time. So how we communicate this to the public is equally important to the science that's been done over the previous 10 or 15 years. So those two things, I think, they're key.
Yeah, okay. Paul, you talked about, I suppose, a lot of the measures that you mentioned, like reducing finishing age, they make financial sense for farmers, but you're not seeing as much buy-in as you may have hoped for various reasons. So that's not enough of an incentive. So what else needs to be done to encourage farmers to reduce their finishing age?
Yeah, well, I suppose, Helen, we should look back over maybe the longer term in terms of what has happened in finishing age. If we go back to 2010, for example, we've seen a very strong reduction in finishing age and largely driven by incentives at industry level and maybe more restrictions in terms of, upper carcass weights and age limits and so on. So there was a response to market signals. So I think, certainly market signals will play a role, but finishing age, I would say, is an all-encompassing trait. It's not a single trait as such. So all of the other factors that are being addressed in other areas of the research programme and the extension programme, health, nutrition, genetics and systems, if we look at those 4 planks, all of those 4 planks are economically beneficial. We know, and very interesting, Suzanne's point around, the opportunities with methane EBVs, which we now have in Ireland as well. If you select heavily on a particular EBV, then obviously you have a compromise just in terms of what your selection pressure is. So it depends how hard you want to push that. Equally on health, we are making a lot of progress, but we can see that there are still opportunities in relation to health. And finally, I'll just make the point on systems. A lot of our farm units are operating animals, maybe not according to very defined systems, and greater information around that, so the animals are targeted into particular systems and are therefore managed in a specific way would have a big impact on that.
Okay, I'm going to go to some Slido questions coming in, and I may come to you with this, Sharon, anonymously inputted. Will the genetic diversity covered in Rumen Gateway be maintained as a public resource and not locked up by companies if they commercialise a product?
Yes, that is the ethos of the project. So they will be put into culture collections and will be open source. The one caveat to that is on the bacterial side, if there are potentials there for direct fed microbials that could tie up hydrogen, for example, and improve the productivity of the animal, then they'll have to go through IP first before they're released. So whomever has who's isolated them have protected them in terms of IP. but then they will go out immediately.
Okay, this is something, Suzanne, I might put to you. If we're talking about breeding for low methane animals, could that further reduce the already low genetic diversity in livestock, potentially threatening their resilience and health? And another question has come in about maybe animal welfare trade-offs if it comes to breeding for lower emission animals.
Yeah, the level at which we're looking at selecting is not going to impact inbreeding in any way, and it's not expected to impact animal welfare. We haven't seen any animal welfare concerns. In actual fact, we see healthier rumens and probably better immunity in our low methane stock. So we're not expecting any sort of trade-offs with inbreeding with a 1% drop.
Okay. And for the panel in general, if we're looking at these reduction measures for methane and nitrous oxide that we heard earlier, how far will they get us towards meeting our targets? And herd size reduction, will that have to be something that's looked at as well?
Do you want me to start, maybe?
Yeah, please.
2030, certainly, the map is there, and we have lots of solutions. Our challenges are 2030 to 2050, and much like New Zealand, Northern Ireland also has a separate methane target of 46% by 2050. So it's certainly that gap, and that is why what we're all doing on this stage and the previous speakers is so important, because that's when the technology that's being developed now is going to come and fill in those gaps.
Okay, does anyone else want to touch on that briefly?
Yeah, I think it's a matter of timing as well. Some technologies are ready to be used now. Some models will come in future years. So I think it's going to be a combination of different technologies for sure, for how we can achieve the targets.
Okay, great. So we're happy to leave it there. Did you want to come in on that?
Yeah, no, well, what I would just say is to add to that, I mean, we have a roadmap, or we have a margin abatement cost curve. We have a suite of technologies available, and that is our focus and should be our focus, to see can we get those adopted. We have the signpost program to try and implement those and deploy those technologies. That is the objective at the moment. Post 2030, new technologies will emerge, and we'll see what happens after that.
Great. Okay, well, I'd like to thank all of our panel, Sharon Huws, Suzanne Rowe, David Yanez-Ruiz and Paul Crosson. Thank you all very much. And so now we'll move on to the best part of the conference, which is lunch. You can all make your way to the foyer. I'm told an hour and 15 minutes for lunch, which means we need you back here. It's 10 to 1. We need you back here at 2:05, and we'll resume the conference then. Enjoy your lunch. Thank you very much.